Before you touch a single reagent, answer one question. What is this extract going to be used for? Because the lysis buffer that works perfectly for a Western blot will destroy an enzyme activity assay, and the DNA storage buffer that's fine for PCR will inhibit a sequencing run.
What we're doing in any extraction protocol is disrupting the cell membrane and subcellular structures to release the macromolecules inside, genomic DNA from the nucleus, proteins from the cytoplasm and organelles, and then isolating the fraction we want in a form that's compatible with what comes next. The biology doesn't end at the lysis step. Everything after is protecting what you already released.
Two separate isolation paths, but they start from the same cell, and if you plan it right, you can run both from the same sample without splitting your material in half.
It is mildly inefficient that most protocols instruct you to discard the fraction you didn't plan for.
3 separate kit purchases, or one TR-isol-based extraction that pulls DNA, RNA, and protein simultaneously from one sample. The math is straightforward. So is the storage requirement, which Kelpie will get to.
Centrifuge. You need one that can hit at least 14,000 times gravity for debris clearance. A basic benchtop microcentrifuge does this. Know the difference between pelleting cells, clearing lysate, and ultracentrifugation for membrane fractions. 3 different speed ranges, 3 different things you're removing.
I set mine up on the bench, room temperature.
Stop! For protein extractions especially, everything runs on ice from the moment you add lysis buffer. Room temperature means your endogenous proteases are active from the first second of lysis. Your proteins are digesting themselves before you can even load a gel.
Proteases are released alongside the proteins you're trying to preserve. At 37 degrees, their activity is maximal. At 4 degrees, it is dramatically reduced, but not zero. This is why the 2-step rule is non-negotiable. Chill all tubes and buffers before you begin, and work on ice throughout. The cold does not stop proteolysis, it slows it enough to give you time.
It is mildly proactive to digest your sample before the experiment begins.
Protease inhibitor cocktail. Goes into your lysis buffer before you touch the cells. Not after. If you add it after a suspension, the damage is already done. Tablets or liquid cocktail either works. PMSF is also widely used, but has a 30-minute half-life in aqueous solution. Add it fresh, not to a stock buffer you prepared yesterday.
Protease inhibitor tablets run around $16 each. A single tablet typically covers 24 to 40 reactions. That is less than 70 cents per experiment. Skipping it and losing the entire protein prep costs considerably more, in both reagents and morning.
Lysis buffer selection is where most people make their worst decision of the day, usually because they grabbed whatever buffer was on the shelf. Reaper is the default in most labs. It works, it's aggressive, it extracts everything. But Reaper has SDS, sodium deoxycholate, and NP40. The SDS alone is enough to denature your protein and destroy any protein-protein interactions you were hoping to preserve.
The choice of detergent determines the physical state of your extracted protein. SDS-containing buffers, like REPA, fully denature proteins by disrupting hydrophobic interactions and unfolding the polypeptide chain. Mild non-ionic detergents, like NP40 or Triton X100, permeabilize membranes while leaving most protein complexes intact. Native lysis, mechanical disruption in a non-detergent buffer, preserves conformation entirely. Each is correct for a different downstream purpose.
Lysis. Chemical lysis. Add your buffer. Incubate on ice for 20 minutes. Vortex gently every 5 minutes. For tough cells or tissue, you'll need mechanical help, sonication or bead beating. Tissue needs either a freeze-thaw cycle first or physical homogenization. Do not add heat to speed this up. There is no recoverable situation after you've heated your protein lysate.
Sonication is efficient, but introduces shear force that fragments genomic DNA. This is irrelevant for protein extraction. You want the DNA sheared so the lysate clears easily, but critical for DNA extraction where you're preserving the molecule. Choose your mechanical method, knowing which fraction you care about. If you want both, you must split the sample before the sonication step.
For simultaneous DNA, RNA, and protein from one sample, trizol is the standard, phenol chloroform-based, 3 phases. Aqueous top layer has your RNA, and with back extraction steps, some DNA. Interphase and organic layer have your proteins. You pull each fraction separately, process each for its downstream use. 1 sample, 3 fractions, nothing wasted.
The separation is driven by differential solubility. At acidic pH, DNA partitions to the organic phase along with proteins. At pH 8, DNA migrates to the aqueous phase. This is why pH control in phenol extraction is not optional. The chloroform improves phase separation and reduces protein carryover. The interphase, which people usually discard, contains the genomic DNA.
DNA precipitation. Add isopropanol to the aqueous fraction, 0.7 volumes. Incubate at -20 for at least 30 minutes. Pellet at 14,000 xg for 10 minutes. Wash the pellet twice with 70% ethanol to remove salt. Air dry for a few minutes. You want no visible ethanol, but do not overdry the pellet. An overdried DNA pellet will not resuspend and you will lose it.
The ethanol wash step is underappreciated. Residual salt from the precipitation buffer inhibits downstream enzymatic reactions. Restriction digests, PCR, sequencing. 2 washes with 70% ethanol are standard. After the second wash, allow complete evaporation of residual ethanol before adding resuspension buffer. Ethanol at even trace concentrations in the final prep will inhibit TOC polymerase.
I didn't add RNase A. I didn't see it in the protocol.
To the DNA prep.
To the DNA prep, yes.
Your genomic DNA prep now contains a significant amount of co-precipitated RNA. Your nanodrop ratio will look fine, probably better than fine, and your yield number will be inflated. When you run it on a gel, you'll see a diffuse smear below the high molecular weight genomic band. That's your RNA. Add RNase A at 20 micrograms per milliliter to the resuspended DNA. Incubate at 37 degrees for 30 minutes, then reprecipitate or column clean.
RNA co-precipitates with DNA under standard alcohol precipitation conditions because both are negatively charged nucleic acids with similar physical properties. A nanodrop reading of 1.8 to 2.0 on 280 is consistent with clean DNA, but also with DNA contaminated with RNA. RNA absorbs at 200 scasm indistinguishably from DNA. The only way to confirm RNA-free genomic DNA is to run it on a gel. A clean gDNA prep shows one tight band at the top of the gel and nothing else.
Resuspend in TE buffer for storage. TE 10 millimolar tris, 1 millimolar ether, pH 8. The ether chelates the magnesium that dinases need to function. That's what gives you long-term stability. If you're going straight to PCR, use nucleus-free water instead. EDTA at 1 millimolar is below inhibitory for most reactions, but strip it out for any magnesium-sensitive enzyme work. Protein extraction, cell pellet, wash it once with cold PBS to remove culture medium. Medium has proteins in it. You don't want them in your lysate. Then add your lysis buffer with inhibitors already in it, on ice. Incubate 20 to 30 minutes. Vortex gently every 5. For adherent cells, scraping and buffer is better than trypsin if you need nuclear proteins. Trypsin removes surface proteins and can cause signaling artifacts.
Quietly. I added the protease inhibitors after. I resuspended first and then added the tablet.
How long between adding lysis buffer and adding inhibitors?
About 15 minutes. I was reading the tablet packaging.
Proteolysis begins within seconds of lysis. The proteases that were sitting in your lysosomes are now free in solution with your proteins, and they have been active for 15 minutes at whatever temperature you were working at.
Endogenous proteases are abundant. Cathepsins, calpins, and serine proteases are all released during cell lysis. Their activity depends on temperature and substrate availability, both of which suddenly become favorable the moment the membrane is disrupted. 15 minutes at ambient temperature is enough time for significant degradation of label proteins, particularly signal transduction components and transcription factors, which have short half-lives by design. Add inhibitors to the lysis buffer before any cell contact. Always.
Clearing spin. 14,000 xg, 4 degrees, 10 minutes. The pellet is your cell debris, unbroken nuclei, and anything insoluble in your lysis buffer. Transfer only the supernatant. Transfer carefully. If you're using Ripper and resuspend any debris, you get gel artifacts and background that takes a gel strip to fix. Your cleared lysate is your starting material for everything downstream.
What remains in the pellet after clearing depends on your lysis buffer. REPA solubilizes most cellular proteins, including membrane-associated ones. A mild detergent like NP40 leaves nuclear proteins in the pellet, which can be an advantage if you want a cytoplasmic versus nuclear fraction. Native buffers leave cytoskeletal proteins and membrane complexes largely insoluble. What you spin away is not always waste.
Sequential extraction. Cytoplasmic fraction first with mild detergent. Nuclear fraction after with a stronger buffer. Is used in chromatin IP, transcription factor studies, and nuclear receptor assays. Commercial kits for this run $40 to $80 per prep. Knowing the principle means you can do it with buffers you mix yourself.
DNA integrity is something nanodrop cannot tell you. Run a gel. Clean genomic DNA shows one tight band at the top of the gel, nothing else. If you see a smear below the band, RNA contamination. If the whole lane smears, shearing during extraction. If there's nothing, you lost the pellet during one of the washes. A gel takes 15 minutes and tells you whether the nanodrop number is real.
Protein quantification by Bradford or BCA gives you total protein concentration in the lysate. This is the number you use to normalize loading across all samples in a Western, ELISA, or activity assay. Do not load by volume. Variation in cell number, lysis efficiency, or protein yield between samples is invisible to volume-based loading and will confound every downstream comparison you make.
Fresh standards every time. Your BSA standard curve from last week is not your BSA standard curve this week. Takes 5 minutes to prepare. Do not skip it to save 5 minutes, and then spend 2 hours trying to explain why your loading looks uneven.
This is the table the protocol sheet never includes. REPA is the right choice for Western blotting, where you need complete protein solubilization and you don't care about conformation. It is the wrong choice for co-immunoprecipitation, where the antibody needs to recognize a native epitope and the complex needs to hold together. It is certainly the wrong choice for enzyme activity assays, where the SDS has already unfolded your enzyme. NP40 covers the middle ground. Native lysis covers everything where conformation is the point.
And ELISA depends entirely on whether you're measuring a denatured antigen or a native one. Most ELISAs use a capture antibody that recognizes native protein. Run your extract in Reaper, and you may denature the epitope the capture antibody is looking for. Check the assay kit instructions for recommended lysis conditions. They usually bury this information in the FAQ section at the back. DNA storage. For PCR and routine genotyping work, -20 in TE, limit freeze thaw to under 20 cycles, it'll be fine for years. For long-read sequencing, Oxford Nanopore PacBio. You need intact high molecular weight DNA. Go to -80. Single-use aliquots, never vortex. Invert gently to mix. Every freeze-thaw cycle introduces nicks. Every vortex step shears. Long-read sequencing will fail on DNA that looks perfectly acceptable on a short-read nanodrop and gel.
The fragility of high molecular weight genomic DNA is underestimated because standard gel electrophoresis resolves fragments in the range of 100 to 10,000 base pairs. Long-read platforms require fragments above 10 kilobases, often above 50. At that size, the DNA is mechanically labile. Pipette shear alone during normal handling generates double-strand breaks. Wide-bore pipette tips and slow resuspension are not optional for long-read applications.
Protein storage. Rule 1. Aliquot before you freeze. A protein lysate you thaw and refreeze 5 times has lost measurable activity by the 3rd cycle for most labile proteins. Rule 2. Glycerol. 10 to 20% glycerol in your storage buffer reduces ice crystal formation and extends shelf life meaningfully, particularly for native protein preparations and anything enzyme-based. Rule 3. -80, not -20. Proteins are not DNA. -20 is not cold enough for long-term protein stability.
Enzyme activity is especially sensitive to freeze-thaw-induced aggregation. The mechanism is ice crystal formation. At -20, water does not freeze completely, and partial ice formation concentrates solutes, increasing ionic strength and driving protein-protein contacts that lead to irreversible aggregation. -80 freezes rapidly and uniformly, which is why snap freezing in liquid nitrogen before transfer to -80 storage is the standard for activity-sensitive preparations.
Diagnostics, research, biobanking, proteomics. The applications all reduce to the same requirement. Know what you're extracting for before you start. The lysis buffer decision is made once, at the beginning, and it commits every downstream step.
From a clinical diagnostic perspective, extraction quality determines assay sensitivity. A genomic DNA prep with residual inhibitors will give false negatives in a PCR-based pathogen detection assay. A protein prep run in an incompatible lysis buffer will give false negatives in an ELISA. Either outcome affects a clinical decision. The extraction is not a preparatory step. It is part of the assay.
An ELISA kit for a standard cytokine panel runs $300 to $400. Lysing the cells in REPA when the kit requires native protein and running the assay anyway costs $300 to $400 in a morning. This is not a rare scenario.
It is mildly instructive that the most expensive part of an experiment is usually the step that happens after the cheap mistake.
I should also mention that for the last 14 months, I have been running parallel DNA and protein extractions from environmental tissue samples collected from sites surrounding 3 industrial facilities my shed overlooks. Unauthorized biobanking, obviously. Nothing institutional about it. Both fractions. Full downstream compatibility testing.
The March batch went in last night. Both fractions, separate tubes. I used native lysis buffer for the protein this time. You said you wanted the enzyme activity data.
It is mildly reassuring that at least one biobank in this building has proper fraction labeling.
The documentation is genuinely quite rigorous.
14 months, and 2 fractions per sample. Everything logged. Sample name, lysis buffer, date, all 3 on every tube, because 6 months from now, when you want to run a new assay on archived material, you need to know how it was lysed. Without that on the tube, you are guessing. And guessing costs you the experiment, or in our case, the entire longitudinal study. That's extraction. DNA and protein from the same cell, stored correctly, compatible with whatever you need downstream. Next Monday, we're in the gel room. DNA electrophoresis, pouring the agarose, loading your samples, and reading every band on that gel, including the ones you didn't plan for, because those are the ones that tell you something went wrong 2 steps back. If you want to call yourself a mad scientist, you need to be able to read a gel without squinting at it for 20 minutes. Method Monday, every Monday.